In this video, we're going to go through some revision notes for Biology 0610, Cambridge IGCSE, for topic 8, transportation in plants. So let's begin with the two types of transport vessels in plants, which are the Xylem vessel and the Phloem vessel. The Xylem vessel transports water and minerals from roots to the stems and leaves, while the Phloem vessel transports food in the form of sucrose and amino acids made by the plant from photosynthesizing to non-photosynthesizing regions into roots and stems. Later on, we're going to learn that phloems transport sucrose and amino acids from photosynthesizing regions called source to non-photosynthesizing regions called sinks. This is just to keep in mind. Both of these vessels are arranged in vascular bundles throughout roots, stems, and leaves. So the place where xylem and phloem both meet is known as the vascular bundle. Okay. Xylem is always towards the upper epidermis, and phloem is always towards the lower epidermis. Let's have a look at the diagram here. So this is a 2D diagram of the cross-section of a leaf. We've discussed the parts of the cross-section of a leaf before in the previous chapters, the palisade mesophyll, spongy mesophyll, air spaces, guard cells, stomata, and etc. But here, all we need to focus on is the xylem vessel and vascular bundles. So this is the xylem vessel towards the upper epidermis, this is the phloem towards the lower epidermis, and the point which they both meet, this is the vascular bundle. Alright? Let's take a cross-section of this leaf and the cells present inside. So in a cross-section of a root stem leaf, we have this leaf. The phloem tissue is towards the outside and the xylem tissue is towards the inside. And the cross-section of a root, we have the root cortex cell. The phloem tissues are concentrated on the outside, while there's one big xylem tissue concentrated in the middle. Okay, so a quick review that we have two types of transport vessels in plants, the xylem vessel and the phloem vessel. The xylem vessel transports water and mineral ions from the roots to the stems and the leaves, while the phloem vessel transports food in the form of sucrose and amino acids to make proteins. These are made by the plant from photosynthesizing regions known as source to non-photosynthesizing regions known as the sink. Both of them are arranged in vascular bundles, and the xylem is always towards the upper epidermis, the phloem is always towards the lower epidermis. In a cross-section, we have a cross-section of a stem and cross-section of a root. In both cells, you will always find xylem on the inside and phloem on the outside. Now let's take some adaptations of xylem vessels. Xylem vessels, the cells are joined with no cross walls to form a long continuous tube to let water move from the roots to the leaves. So, adaptation of the xylem vessel, the top and bottom, let's have a look at this diagram here. We have no top and no bottom walls. This allows a long continuous tube to let water move through the leaves freely. The cells are dead. They don't have any cell contents inside it. This allows free passage of water. So if you have a look inside the cell, you will not find any mitochondria, nucleus, ribosomes, and etc. This is to allow free passage of water. The outer walls are thickened with lignin. Lignin is a structure that strengthens the tubes and supports the plant. This structure outside, this is lignin. It's holding the xylem vessel together, and it provides strength and support to the plant. Next is the root hair cells. Root hair cells are single-celled extensions of epidermis cells in a root. Root hair cells grow between soil particles and they absorb water and minerals from the soil. They absorb these water and minerals from the soil because they have to transport it to the plant via translocation. Water passes into the root hairs via osmosis through the root cortex and into the xylem vessels, because the soil water has a higher water potential and the cytoplasm of the root cell has a low water potential. So what's happening is that as we put water into the soil, you're putting water from high water potential, and the water is going to go from high water potential to low water potential into the root cortex cells, into the phloem. Once it goes into the phloem, the root cortex cell is going to transport this into the xylem via osmosis, and the xylem vessel is going to transport this water and mineral ions throughout the whole plant. Root hair cells increase the surface area of cells, which increases the rate of absorption of water by osmosis and mineral ions by active transport. So root hair cells perform both osmosis and active transport, while xylem only performs osmosis to move and transport water. This is the root structure diagram. So here we have soil, and we have root hair cells. As we put water, water is going to move into the root cortex and via osmosis into the roots, then to be transported to the plant via xylem. The pathway of water into and across the roots. This is a very important one because we have a lot of questions for this in paper 2, paper 4, and paper 6. So once water gets into the xylem, it's carried up to the mesophyll cells. The pathway is root hair cell, then root cortex cell, then xylem, then leaf mesophyll cells. So we put water inside the soil. This is the soil particles. It has high water potential over here. The water moves from high water potential to low water potential into the xylem vessels via root hair cell, via root cortex, into xylem vessels to be transported to the plant. From high water potential to low water potential. High water potential in the soil and low water potential in the root cortex cells. So this way we have covered the basics of xylem and phloem. Now we're going to take our first experiment in this chapter, which is investigating water movement in plants. This is a very easy one. So first, we place a plant into a beaker of water with stain or food coloring in it. So you have a beaker of water, and this beaker of water contains food coloring. Then you place a plant inside it. After a few hours, leaves of the plant turn same color as the dyed water, proving water is being taken up by the plant. So the plant contains xylem vessels which uptake water. Now, if you're going to put food coloring inside the beaker of water, the color of the water is going to change. As you put the plant inside, the xylem vessels are going to absorb this water from this food coloring, and it's going to change the color of the plant. So to finalize the experiment, if a cross-section of a leaf is cut, only certain areas of the stalk is stained with the color of the dyed water, showing water is being carried by only specific xylem vessels through the stem. So this is the experiment. We have 3 plants in sunlight for several hours. The first one is placed in water, second one is placed in red food dye, and third one is placed in blue food dye, or methylene. After a period of time, we discovered that the first one has no stain because it absorbed just water without any dye coloring, while the other 2 are stained by the color of their dyed water. That's an easy experiment. Let's move on to transpiration. Transpiration is something that occurs in xylem vessels only. This is the loss of water vapor from plants' leaves by evaporation of water at the surface of the mesophyll cells, followed by diffusion of water vapor through the stomata. So water vapor is being lost by plant leaves by evaporation at the surface of the mesophyll cells, and this evaporation of water is followed by diffusion of water vapor through the stomata. Why do plants need transpiration? Plants need transpiration to transport mineral ions. They need to keep leaves cool by using up heat to convert water, H2O, into vapor and cool down the plant. The water they receive is going to be converted into vapor and cool down the plant, just like how humans sweat to cool themselves down. And last but not least, plants need transpiration to provide water to the leaves for efficient photosynthesis and to keep the cells turgid and support the structure of the plant. Because as water moves into the plant cell, the cell membrane starts to expand against the cell wall and makes the plant turgid and rigid. Water travels up the xylem from roots into the leaves of the plant to replace water that has been lost due to transpiration. These are some facts about evaporation in plants. Evaporation takes place from the surface of the spongy mesophyll cells. This is by default because the difference between evaporation and boiling is both of them are changing of the liquid state into the gaseous state. But evaporation only occurs at the surface where the highest energy particles escape, while boiling occurs all over the liquid. Evaporation occurs rapidly when the stomata opens, as high energy particles rapidly escape. Mesophyll cells, this is just for general information, but it's not required to know in this chapter. There are cells with interconnective air spaces in between, and the stomata create a large surface area. So this is the xylem in transpiration. This is the xylem vessel, and this is the leaf. The xylem contains a continuous column of water with no top and bottom walls, and no cell structures to allow free movement of water during transpiration. Water is moving up the xylem vessels or being uptaken by the xylem vessels to be transported into the leaves. And from the leaves, it is evaporated outside. Movement of water in the xylem is only one directional, unidirectional from the roots to the leaves. It only moves up, while movement of water in phloem is in many different directions or bidirectional. It moves up and down. Now let's head on to our next experiment. Our next experiment is investigating the effect of temperature and wind speed on transpiration rate. How does temperature and wind speed affect the transpiration rate? Temperature and wind speed over here are our independent variables. These are the variables that we are going to be investigating. So these are the variables that needs to constantly be changed during the experiment, while the transpiration rate is our dependent variable in this experiment, the variable that needs to be measured. So let's undergo the steps of this process. First, cut a shoot of a plant under water and place it in a tube. So we have a plant, we cut a shoot of this plant and place it in a tube. Set up the apparatus upright by using Vaseline to seal any gaps. So the apparatus, we seal it with Vaseline to seal any gaps and prevent air loss. After that, we dry the leaves of the shoot, and then we remove the capillary tube from the beaker of water to allow single air bubble to form, and then place the tube back into the water. So after we've used Vaseline to seal any gaps, we remove the capillary tube from the beaker of water. This allows one single air bubble to form. As soon as the air bubble is formed, we place the tube back into the water, and we start setting up the environment for the factor that we're investigating. This allows the plant to adapt to the new environment for 5 minutes. So once we place the tube back into the water, we set up an environment for the factor we're investigating, whether it's temperature or wind speed. And we let the plant adapt to it for some time. Now we start recording the location of the air bubble, and then leave it for a set period of time. So record the location of the air bubble initially where it was, and leave it for a set period of time. Then record the end location of the air bubble. Change in wind speed or temperature, whichever factor is being investigated, and reset the bubble by opening the tap below the reservoir. So first we record the location of the air bubble, initial location and final location. After recording it, we change whichever factor we are investigating, whether we are investigating wind speed or temperature, as the independent variable. And by the end of this experiment, we are going to reset the bubble by opening the tap below the reservoir and repeat the experiment for validity. The further bubbles travels in the same time, the faster the transpiration, and vice versa. So the distance of the bubbles is directly proportional to the transpiration rate. This is a table to show the transpiration rate factors. The first factor is wind speed. If wind speed is high, we have more airflow to remove water vapor from the air surrounding the leaf, which increases water loss. So this is directly proportional. Humidity. If humidity is high, more water is lost, and if humidity is low, less water is lost. so directly proportional. Temperature. If temperature is high, at higher temperatures, particles have more kinetic energy and transpiration occurs at a faster rate, so it's also directly proportional. This is the illustration diagram to show our second experiment, which was about investigating the effect of temperature and wind speed on the transpiration rate. The equipment's apparatus is not necessary to be memorized, but just to explain, we have the reservoir over here, the one that we seal with Vaseline. This is a cut chute placed under water. This is a lamp to investigate light intensity as the independent variable. This is the beaker of water. This is the volume scale to measure the distance of the air bubble, and this is the capillary tube. So first we record the distance of the bubble at the start of the experiment and leave it for a set period of time using a clock, and then record the end location and calculate the distance between traveled. By the end, we reset the air bubble using the tap reservoir and repeat the experiment for validity. The only equipment required for you to memorize in this experiment is the potometer, capillary tube, and graduated scale. Potometer is an apparatus to investigate the effect of environmental factors on the rate of transpiration. Capillary tube is an apparatus to prevent bubbling moving. And graduated scale is to allow bubble movement to be read. The investigation of such environmental factors, in our syllabus we have temperature and wind speed and humidity. Temperature and wind speed are both directly proportional. The test of temperature is done in a cold and warm room, and the result, as the temperature increases, transpiration increases. Wind speed test is done by using an electric fan to mimic different wind speeds as the independent variable, and the result shows that as the wind speed increases, the transpiration rate also increases. And with that, we're done with everything regarding the xylem and transpiration in plants. The only left for transpiration is the transpiration stream, and then we move on to translocation. Before moving to transpiration stream, a small key point to understand is wilting. Wilting is when more water evaporates from the leaves of the plant than is available in the soil to move into the root by osmosis. So plants have water available in the soil that moves by osmosis, but what happens during wilting is that more water is evaporating by the plant than what is already available in the plant. When cells of the plant are not full of water, they lack H2O. The strength of the walls cannot support the plant because the plant cells are losing water via osmosis and they are becoming plasmolized. Hence, during wilting, the stomata will close to reduce the heat absorption, evaporation, and diffusion of water. Transpiration stream. Transpiration stream is a continuous movement of water through the xylem vessels from roots to leaves via stem. So we have a plant with roots and leaves. Transpiration stream transports water from roots to leaves via stem. Okay, how does this happen? How does water transport itself upwards in the xylem, defying gravity? This is because of cohesion forces or cohesive forces. Cohesion is the attraction force of water molecules between each other, holding the hydrogen-water bond together. So we all know that water is held together by hydrogen bonds, and these hydrogen bonds attract each other. So this attraction force between water molecules between each other holds the hydrogen-water bond together, and this creates a continuous column of water up the plant because each individual water molecule is using cohesive forces between the hydrogen bonds to pull upon the one below it, and this way water is pulled up through the plant. Transpiration produces a tension pull because the forces are creating a continuous column of water and the xylem vessels by the leaves. If transpiration rate from the leaves increases, water molecules pulled up the xylem vessels faster, so it is directly proportional. This is a quick illustration. We have the roots. Water moves from the roots to the stems into the leaves via transpiration stream using cohesive forces to attract H2O to each other. This is the generation of the transpiration stream. So we have xylem vessels with lignified walls over here, and adhesion is occurring between the cellulose and water inside the xylem vessels. This uses water molecules, and this is the cellulose molecules. And at the same time, we have cohesion occurring inside the water molecules. This creates tension to enable water to be pulled upwards. This is the vacuole, this is the cytoplasm, this is the cell wall, chloroplast, and evaporation of water is happening here due to high heat energy. This is the stomata and guard cells. Translocation. Translocation is the transport of sucrose and amino acids in the phloem from regions of production, which is the sucrose, where food is made, to regions of storage, which is the sink, where food is not made. So translocation occurs in the phloem, like transpiration occurs in the xylem, translocation occurs in the phloem. From regions of production, photosynthesizing regions to non-photosynthesizing regions. Soluble products of photosynthesis are sugars, which is sucrose and amino acids. Both sucrose and amino acids are transported in the phloem via translocation. So during translocation depends on the weather or the season. Okay, so during summer, what we have is we have a plant with roots and present leaves. During summer, since the leaves are present, the source is the leaves and the sink is the root. So amino acids and sugars transport from source to sink, from leaves to the roots. While in winter, it's quite the opposite. We have a plant with roots, but growing leaves. So leaves are quite much absent. In this case, the source is the roots and the sink is the leaves, and water and sugars and amino acids are transported from the source to the sink, from the roots to the growing leaves.